Autism-linked gene in placenta. Greetings, friends of Ai4Autism. In this episode, we're taking a look at the Spectrumnews article, DNA tags in placenta point to possible autism-linked gene. So let's begin. A gene not previously linked to autism alters the expression of other genes associated with the condition, according to new analysis of DNA in the placenta. The placenta's genome comes from the fetus and has a unique pattern of methylation, the suite of chemical tags called methyl groups that attach to DNA to regulate gene expression. The pattern may reflect brain development and a child's chances of having autism, some research shows. In the new work, researchers analyzed methylation levels across an entire genome in 204 placentas from births of children who have an autistic older sibling. Such children, known as baby sibs, have an increased chance of having autism. A stretch of DNA called 22q13.33, from here on I'll call it 22q, which has not previously been linked to autism, tends to be less methylated in placentas from the births of children who are later diagnosed with the condition, the researchers found. The findings, along with previous work on the placental methylome, show that the organ may mirror methylation patterns and gene expression levels in the fetal brain, says lead investigator Janine LaSalle, professor of medical microbiology and immunology at the University of California, Davis. We don't exactly know why that is, LaSalle says, but it's exciting in that the placenta could be a good surrogate for what happened in the brain during development. The study demonstrates the importance of studying the off-ignored placenta to understand brain development, says Anna Penn, Associate Professor of Pediatrics at Columbia University, who was not involved in the work. If we're going to understand diseases that we have identified as potentially having an origin in fetal development, we can't continue to ignore the role of the placenta, says Penn, who coined the term neuroplacentology to describe the study of the placenta's effect on fetal brain development. Maybe someday we'll respect the placenta enough not to throw it away. LaSalle's team identified 134 genomic regions spanning 183 genes, many of which are linked to autism, that show different methylation patterns in autistic versus non-autistic children. 12 of the regions, all with unusually low methylation levels, are located in 22q. Though this stretch of chromosome 22 has not been previously associated with autism, it neighbours 22, home to SHANK3 and other genes connected to autism, intellectual disability, or neuropsychiatric conditions. Knowing more about what may be happening in this region will potentially further step in the autism field. Let me read that again. Knowing more about what may be happening in this region will be potentially a further step for the autism field, says study investigator Yihu Zhu, an applied bioinformatics scientist at 10x Genomics in Pleasanton, California, who completed the study as part of her doctoral research in LaSalle's lab. The region contains no previously identified genes, but one sequence encodes a peptide expressed in the brain, placenta, and ovaries, the team found. The gene's expression increases after neurons mature, and in response to oxidative stress, cells derived from embryonic brain tissue revealed. The researchers named the gene neural hypoxia-inducible placenta-associated, or NHIP. Placentas from autistic children have lower levels of NHIP than those from non-autistic children. The findings were published in Genome Biology in February. A common structural variant in 22q occurs more often in autistic than in non-autistic children's placentas, the team found. A similar trend emerged from an analysis of post-mortem cortical brain samples from 27 autistic and 30 non-autistic children and adults. Brain samples with altered NHIP expressions also showed expressions differences in more than 500 genes, including low levels of CHD8, one of the genes most strongly linked to autism. In fact, this is something we touched on in one of our earliest videos. For 77 of the placentas, LaSalle's team had data on women's use of prenatal vitamins which contributed molecules needed for methylation. Those who reported taking vitamins in the first month of pregnancy delivered placentas with higher levels of methylation in 22q than those who did not. Large studies could probe whether carriers of the structural variant should take even higher doses of prenatal vitamins than what is currently recommended, Zhu says. The findings demonstrate how genes and the environment can both contribute to autism, says Magdalena Jenica, Assistant Professor of Psychiatry at the Icahn School of Medicine at Mount Sinai in New York City, who was not involved in the work. You have genetic and environmental factors that converge in the same biological processes, Jenica says. I think that's really cool. The sample size is small, Penn says, though studies of placentas don't tend to get much bigger. The researchers' sophisticated multi-step approach lends weight to the findings, she says. The use of techniques to sequence methylation across the entire genome rather than only certain areas also helps, Jenica says. It's a very elegant study. Comparing methylation across groups rather than in individuals may miss rare changes that contribute to autism in only some people, Jenica says, similar to how some rare gene mutations occur in only a small subset of autistic people. Using methods to identify such rare conditions could turn up more methylation factors that contribute to the condition. LaSalle says that she next plans to more carefully characterize NHIP's function and determine if the NHIP peptide could be used therapeutically. If you managed to listen from start to finish, thank you very much. That was quite the mouthful, both in technical terminology and other things too. And thank you very much for listening, if you have endured that. What are your thoughts on this? It's quite exciting, I think, when it comes to identifying the possible link to autism and the environment in the placenta and beyond. What are your thoughts? Post your comments below. Thanks for watching, and remember, love, life, and AI.